Hello everyone, welcome to today's webinar, Advanced Imaging for PK and PD Analysis to Support Immunodermatology Drug Development. Thank you all for taking time to join us. My name is Morgan from IBM Technology. In today's session, we will explore how advanced intravital imaging can support PK and PD evaluation in immunodermatology drug development. Using psoriasis and atopic dermatitis models as case studies, we will demonstrate how longitudinal intravital imaging enables quantitative assessment of immune cell infiltration, vascular and collagen remodeling, and drug penetration. These imaging approaches provide variable insight into tissue-level therapeutic responses, target engagement, and preclinical candidate evaluation. Today's presentation will last approximately 40 minutes, followed by a 10 minutes Q&A session. So if you have any question during the presentation, please feel free to submit them anytime using the Zoom Q&A panel or chatting box. Also, IBM Technology offers a variety of preclinical CRO service packages covering skin, brain, retina, and many other research applications. If you'd like to learn more, please check the Resource tab below in Zoom to download our CRO package flyers. Now, it is my pleasure to introduce today's speaker. Costa Yoon is a technical application specialist at IBM Technology. Holding a master's degree and extensive hands-on experience, he specializes in animal surgery and experimental procedures for advanced intravital in vivo imaging studies. So Costa, if you're ready, please go ahead with your presentation.
Hello everyone, and thank you for joining us today. My name is Costa Yoon from IBM Technology. Today's webinar is titled Advanced Imaging for PK/PD Analysis to Support Immunodermatology Drug Development. In this session, I will introduce how intravital microscopy can be applied to drug development for immunodermatological disease. Specifically, I will present our research on PK/PD analysis in mouse models of psoriasis, atopic dermatitis, and hair follicle imaging for alopecia drug development. And lastly, wound healing studies enabled by intravital imaging. Skin disease affect about 25.7% of the global population, roughly 1 in 4 people. Among these, immunodermatological diseases are caused by dysregulations of immune system. They represent a major group of skin disorders associated with a substantial chronic burden and long-term treatment needs. Representative disease includes psoriasis, atopic dermatitis, alopecia areata, and vitiligo. In particular, psoriasis and atopic dermatitis are widely recognized as key targets due to their high prevalence and large therapeutic markets. So let me briefly introduce the key characteristics of this disease. Psoriasis is characterized by immune dysregulation and excessive proliferation of keratinocytes. As a result, the skin develops thick, silvery white scales, and uncontrolled inflammation leads to blood vessel dilation and visible erythema. On the other hand, atopic dermatitis is associated with skin barrier disruption and abnormal immune responses. It marked by severe itching, oozing from affected skin, and the subsequent formation of crusts. From 1990 to 2021, the number of patients with psoriasis and atopic dermatitis increased by approximately 1.8-fold and 1.2-fold respectively. These trends clearly indicate that the global burden of immunodermatological disease continues to rise. With the increasing number of patients, companies are actively developing new treatments with improved efficacy, safety, and dosing convenience. Driven by these unmet medical needs, the therapeutic markets for both psoriasis and atopic dermatitis are expected to expand significantly. Next, I would like to briefly discuss how treatments for psoriasis and atopic dermatitis have evolved. In the past, topical corticosteroids and immunosuppressants were commonly used to broadly reduce inflammation and control symptoms. In psoriasis, TNF-alpha inhibitors were introduced as a new treatment option. More recently, therapies have been developed to target key molecules in the disease process, including IL-17 and IL-23. Similarly, in atopic dermatitis, drug development has actively focused on key signaling pathways, such as IL-4 and IL-13. Despite the rapid progress in immunodermatology, there are still several challenges in developing effective therapies. The first challenge is the skin barrier. Topical treatments are widely used because they can be applied directly to the affected area and reduce systemic side effects. However, the stratum corneum acts as a strong physical barrier, making it difficult for drugs to reach their target tissue, such as dermis. To overcome this limitation, a variety of delivery strategies, including nanoparticles and penetration enhancers, are being actively explored. However, conventional ex vivo methods cannot fully recapitulate the complex physiological environment of living tissue, including dynamic inflammation, vascular responses, and tissue remodeling. Therefore, to evaluate the PK of a drug accurately, we need to know how much of it reached the target tissue, in vivo, and how long it remains there. The second challenge is the rising efficacy standards in clinical development. For example, in psoriasis, the expected level of therapeutic efficacy has continued to rise. PASI is a key clinical index that measures the affected skin area and the severity of the lesion. In the early 2000s, 75% improvement in PASI was widely used as a main efficacy endpoint. Today, many newer therapies aim for 90% or even 100% improvement, meaning a much greater improvement in skin lesions. This shift reflects improvements in therapeutic efficacy, as well as the growing need for pharmaceutical companies to develop more competitive therapies. As clinical expectations continue to rise, conventional ex vivo analysis alone are becoming insufficient to bridge the gap between preclinical findings and clinical outcomes. To improve clinical predictability and support better candidate selection, in vivo PK and PD data are becoming increasingly important. So, intravital microscopy is a high-resolution technique that enables cellular-level visualization in living animals. Because the same animal can be imaged rapidly over time, longitudinal studies can be performed in the same animal, which allow us to minimize inter-animal variability and evaluate treatment responses more consistently. The platform enables PK analysis of drug penetration depth, distribution, and retention. It also supports precise PD analysis by tracking changes over time, including vascular dilation, immune cell infiltration, and collagen remodeling. In summary, intravital microscopy complements conventional ex vivo analysis by providing in vivo PK/PD data. As a result, it improves clinical predictability and supports more reliable go and no-go decision in preclinical drug development. So, how is intravital skin imaging actually performed? Our platform supports imaging of more than 30 different organs using dedicated organ-specific accessories. For skin imaging, we primarily use staging platform, thermoglass cover, and dorsal skin fold chamber. Depending on the experimental object, intravital skin imaging can be performed in different ways. For long-term longitudinal studies, dorsal skin fold chambers or imaging windows can be implanted to provide stable access to the tissue. For shorter experiments, single-session imaging can be performed without any chamber implantation. For skin imaging, we mainly use dorsal skin and ear skin. The dorsal skin provides relatively large imaging area, and it is positioned on a staging platform and gently pressed with a thermoglass cover. We then image the area in direct contact with the glass. For long-term studies, a dorsal skin fold chamber can be implanted to provide better access to deeper tissue layer as one side of the skin is removed. This model is also widely used for melanoma and other skin cancer studies. Ear skin is easier to mount and can be gently planted against the glass surface for stable imaging. The video on the right side shows immune cell recruitment at 0, 3, and 6 hours after injection of an inflammatory stimulus. Using this approach, we can monitor dynamic immune responses over time and apply the platform to psoriasis, atopic dermatitis, hair follicle imaging, wound healing, and other areas of skin research. And intravital imaging is primarily based on fluorescence signals. For most targets, fluorescently labeled antibodies are administered intravenously to visualize specific structures or cell population. Because a defined amount of antibody is injected according to the body weight of each animal, highly consistent labeling can be achieved. For example, blood vessels can be labeled to visualize tissue architecture, while immune cells population, including neutrophils and T cell, can also be specifically labeled. Furthermore, therapeutic agents themselves can be fluorescently labeled to evaluate their biodistribution and skin penetration in vivo. This approach is compatible with wide range of therapeutic modalities, including peptides, antibody, and exosome. Alternatively, reporter mice can be used to visualize targets that are difficult to label using antibodies alone. In the example shown in the upper right side, blood vessels are labeled with fluorescent antibody, while immune cells are visualized using reporter mouse. Finally, in two-photon imaging mode, collagen can be visualized without additional labeling because of its intrinsic second harmonic generation signal. As shown here, this signal helps us identify the collagen-rich dermis and determine how deeply the drug penetrates into the skin. Next, I'd like to present several application cases of our platform. This includes PK and PD analysis in mouse models of psoriasis and atopic dermatitis, two representative immunodermatological diseases. I will first present the PK and PD analysis performed in psoriasis mouse model. First, let me briefly explain the pathological mechanism of psoriasis. In response to various stimuli, activated dendritic cells stimulate Th17 cells, leading to increased IL-17 production. And IL-17 causes stromal cells to release chemokines, which attract neutrophils and other immune cells to the affected tissue. This excessive inflammation leads to blood vessel dilation, tissue damage, and extracellular matrix remodeling. Therefore, IL-17 is considered a key driver of psoriasis, promoting immune cell infiltration, vascular changes, and tissue damage. So in this study, we evaluate the therapeutic effects of IL-17 neutralization over time in a psoriasis mouse model. The psoriasis model was induced by applying imiquimod cream once daily for 4 days. Saline, or IL-17 neutralizing antibody, was administered once daily by intraperitoneum injection at 1 mpk. To visualize neutrophils consistently, we used fluorescent reporter mouse and performed imaging of the dorsal skin. Imaging was conducted before treatment on day 1, day 2, day 3, and day 4 after treatment. To access vascular dilation, blood vessels were labeled with a fluorescent antibody. As mentioned earlier, neutrophils were visualized using reporter mouse, and collagen and dermal layer were visualized using second harmonic generation imaging, SHG. So figure A shows gross image of skin lesion after psoriasis induction with imiquimod and treatment with either saline or IL-17 neutralizing antibody. The orange color lesions area was visibly reduced in treatment group. This macroscopic improvement was also consistent with the intravital microscopy result. In these images, the white signal represents collagen and indicates the dermal lesion, while the green signal represents blood vessels. In the control group, vascular dilation became apparent from day 3 and was maintained through day 4. In contrast, although some vascular changes were observed in the treatment group on day 3, overall vascular structure and vascular diameter remained more stable than in the control group. This can be interpreted as a consequence of reduced inflammation following IL-17 neutralization. Quantification analysis of the fluorescence image show that vessel diameter increased to more than 10 micrometer in the control group. We also evaluate changes in the collagen-associated SHG signal. In the control group, the SHG signal gradually decreased as psoriasis progressed. In contrast, the signal declined more slowly in the treatment group. Because SH signals are typically strong in the collagen-rich dermis, this result suggests that suppression of the immune response may have reduced inflammation-associated disruptions of the extracellular matrix. Next, we examine neutrophil infiltration, which is another major pathological feature of psoriasis. The dot-like signals represent individual immune cells. In the control group, neutrophil infiltration became prominent from day 2 and progressively increased over time. In contrast, the treatment group showed fewer infiltrating immune cells, particularly during the later stage on day 4. This effect can be explained by reduced chemokine production after IL-17 neutralization, leading to decreased recruitment of neutrophils into the tissue. To summarize, compared with the control group, treatment groups show reduced vascular dilation from approximately day 3 and reduced neutrophil infiltration by day 4. This finding indicates that IL-17 neutralization effectively regulated the inflammatory responses. The slower reduction in the SH signal also suggests that tissue damage and extracellular matrix disruption were less severe in the treatment group. Together, these results demonstrate that intravital microscopy can be used to evaluate multiple therapeutic responses parameters in the real time within the same living animal. The next study was designed to evaluate the skin penetration of the topical therapeutic agent in a psoriasis mouse model. ST3 is a key regulator of inflammatory signal, and its inhibition has shown therapeutic potential in psoriasis. However, topical delivery of ST3 inhibitors can be limited by thickened epidermis and altered skin barrier found in psoriatic lesions. To improve delivery to the target tissue, the ST3 inhibitors was incorporated with discoidal lipid nanoparticles, which are known to enhance transsubcutaneous penetration. The psoriasis model was induced using imicomycin cream as in the previous studies. A free ST3 inhibitor and DNP formulated ST3 inhibitor were topically applied to the ear skin, and imaging was performed after 6 hours. The therapeutic compound was conjugated with FITC to visualize its distribution and penetration, while the collagen-rich dermis was identified using the SHG signal. This slide shows two photon images of psoriatic ear skin after topical application of either the free drug or DNP formulated drug. The red signal represents collagen in the dermis, while the green signal represents the fluorescently labeled drug. In the figure A, a very little drug signal was detected in the SH positive dermal region following application of the free drug. In contrast, after application of the DNP formulated drug, a strong drug signal was clearly observed within the dermis. Figure B shows the distribution of the drug at different imaging depths. From the depths of approximately 24 micrometer, almost no free drug signal was detected. The cut view image also demonstrates that the DNP formulated drug penetrates substantially deeper into the skin. Quantitative analysis based on the fluorescence intensity showed that the free drug signal was no longer detectable beyond the depth of approximately 20 micrometers. In contrast, the DNP formulated drug remained detectable in deeper tissue layer. In summary, two-photon imaging confirmed that the DNP formulation improved drug penetration in actual psoriatic skin and enabled the therapeutic agent to reach the collagen-rich dermis. Next, we evaluate the therapeutic efficacy of the drug. Psoriasis was induced in 4 groups, excluding the healthy control group. The animals were treated with distilled water, DNP without therapeutic activity, and the DNP formulated drug or topical steroid as a positive control. The DNP formulated drug group showed clear improvement in the growth appearance comparable to positive control because epidermal thickening is a major feature of psoriasis. We also evaluated H&E staining and epidermal thickness. Both analyses show significant improvement compared with the control treatment groups. In addition, key psoriasis-related cytokine and inflammatory markers were reduced in a DNP formulated drug group. These results suggest that the drug penetrates into dermis and improves the regions by suppressing C3-mediated inflammatory responses. Next, I will show data demonstrating drug penetration in atopic dermatitis, along other representative immunodermatological disease. This study was conducted to determine whether topical therapeutic agent penetrates within the skin of an atopic dermatitis mouse model. Topical steroids are widely used for atopic dermatitis. However, because they broadly suppress immune responses, long-term use may increase the risk of local adverse effects and infection. And excessive production of reactive oxygen species, ROS, by epidermal keratinocyte is known to contribute to the pathology of atopic dermatitis. Therefore, bilirubin has attracted attention as a potential non-steroidal therapeutic agent because of its strong antioxidant and anti-inflammatory properties. However, native bilirubin has poor stability and is difficult to disperse uniformly in topical formulation. To address this limitation, a more stable synthetic bilirubin isomer was conjugated with PEG. The PEG bilirubin conjugate spontaneously self-assembled into nanoparticles in an aqueous environment, improving its stability and dispersion within the formulation. The nanoparticles were then mixed with hyaluronic acid, which increased the viscosity, so the formulation remained at the application site for longer and also provided a moisturizing effect. The study used NCNGA mice, which are susceptible to atopic dermatitis-like inflammation. The skin barrier was disrupted using sodium dodecyl sulfate, SDS, followed by repeated application of an ointment containing house dust mite extract. After topical drug application, penetration was monitored at multiple time points, and fluorescently labeled drug formulation was used to visualize drug distribution, while SH signal was used to identify collagen and dermal layer. Figure A shows image acquired every 20 minutes from right after applications to total of 100 minutes. Over time, the drug signal gradually entered the skin through the hair follicle. To evaluate long-term penetration, imaging was also performed at 0 and 20 and 12 and 24 hours after application. At these faster time points, clear accumulation and penetration of the drug through appendageal route were observed. The penetration depth was quantified by measuring the deepest tissue layer in which the fluorescence signal could be detected. Significant differences in penetration depths were observed across the different time points. And 20 hours after application, the skin was examined at different depths. The drug signal was detected down to approximately 80 micrometers. These results indicate that the drug primarily penetrates through the hair follicle and reach both the epidermis and dermal regions of the atopic skin lesion. The therapeutic effect of the formulation was subsequently evaluated using ex vivo analysis. With the exception of healthy control, atopic dermatitis was induced in the remaining 5 groups. The animal received PBS, dexamethasone as the positive control, and hyaluronic acid alone, or bilirubin formulation at 2 different doses. In the high dosage group, RS level, T-cell infiltration, and major atopic dermatitis associated cytokines were reduced to level comparable to positive control. First images also show substantial improvement of the lesions in the high-dose topical treatment group. Consistent with these findings, H&E staining show that the epidermal structure become more similar to that of healthy skin. In contrast, the low-dose treatment group showed no clear therapeutic benefit. This result suggests that bilirubin may require a threshold dose such as 60 microgram to produce meaningful therapeutic effects. And the therapeutic effect may result from drug penetration into both epidermis and dermis. In the epidermis, bilirubin may reduce RS production, while in the dermis, it may suppress inflammatory responses. So intravital imaging allows us to identify the tissue layers reached by the drug, providing special PK information to help explain where the therapeutic effects occur. In addition to psoriasis and atopic dermatitis, our platform can be applied to many other skin research. I will briefly introduce examples of hair follicle imaging and wound healing analysis. First, I'd like to show you an example of hair follicle imaging. Hair loss research is another field in which active drug development is currently taking place. This experiment was designed to visualize hair follicles and the surrounding stem cell niche, which contains the stem cell responsible for hair regeneration and the microenvironmental signals that regulate them. We used H2B EGFP reporter mouse, in which GFP is expressed in the nuclei of cells. This allows us to visualize individual cells around the hair follicle and identify microstem cells based on their location. Alopecia areata is caused by abnormal immune response, directly directed against the hair follicle. By combining the approach with the immune cell labeling strategies shown earlier, the platform could be used to simultaneously evaluate immune cell infiltration around the follicles and changes in the follicular stem cell niche. Next, I will introduce an example of wound healing analysis in vivo. This study evaluated wound healing longitudinally at the whole animal level. Lymphatic vessels play an important role in wound repair by removing cellular debris. To visualize them, we use PROX1 EGFP reporter mouse, which express a fluorescent signal in the lymphatic vessels. A needle wound was created in the ear skin, and the same tissue region was imaged on day 0, day 5, and day 9. The loss of lymphatic vessel signal after wound induction was marked with a white dotted line. We then monitor the recovery of lymphatic signals along the wound boundary. On day 5, newly formed lymphatic vessel signals appeared along the wound margin. By day 9, additional lymphatic vessel structures have developed and extended further into the recovering region. This finding can be interpreted as lymphatic remodeling and regeneration associated with wound healing. Based on the research cases presented today, we provide an integrated services for skin research. We develop models of psoriasis, atopic dermatitis, and skin cancer, and others. We also conduct intravital microscopy-based preclinical CRO studies and quantitative data analysis. And for PK analysis, intravital imaging allow us to evaluate the skin penetration and biodistribution of therapeutic agent designed to overcome the skin barrier. In addition, SHG signal enables label-free visualization of the dermis, allowing us to determine drug penetration depths and localization more accurately. For efficacy assessment, we can track changes in the same animal over time. This enables precise analysis of key pharmacodynamic parameters, including vascular dilation and extracellular matrix remodeling and immune cell infiltration. Overall, our in vivo imaging platform complements traditional skin research method. It provides detailed PK and PD data needed to improve clinical feasibility and support more informed drug development decisions. So that brings us to the end of today's webinar, and thank you for attention. I will be happy to answer any question. Thank you.
Thank you, Costa, for the great presentation. So let's begin the Q&A session. So our first question is, does attaching a fluorescent label to a drug affect its efficacy or biodistribution?
Oh, yeah, thank you for your question. Regarding this question, fluorescent labeling may alter a drug's molecular weight. This could potentially affect its efficacy or biodistribution. Therefore, we generally recommend evaluating PD using the unlabeled drug and conducting PK analysis separately using the fluorescently labeled drug. For PK studies, including an appropriate control group, enables relative comparison and provides a more reliable assessment of the drug distribution.
Okay, and the second question is, what is the maximum imaging depth of intravital microscopy?
Oh, yeah. Actually, the imaging depth depends on the animal model and degree of inflammation. But in general, we can image up to approximately 100 micrometer in confocal mode and 200 micrometer in two-photon modes. And within two-photon imaging at this depth, we can clearly visualize dermal collagen through second harmonic generation signal. This allows us to access drug penetration into the dermis.
Thank you, Costa. Thank you everyone for all the questions. A recording of today's webinar will be available soon on our YouTube channel. So if we weren't able to answer your question during today's Q&A session, or if you have any additional questions afterward, please don't hesitate to contact us at cro1@ibmtech.com. As I mentioned earlier, if you're interested in our preclinical CRO service packages, please check out the CRO flyer available in the resource tab at the Zoom. Whether you're working on skin, brain, retina, or other research applications, we'll be happy to discuss how we can support your project. You can also reach us through the Contact Us page on our website. Thank you again for joining us today. We truly appreciate your participation, and we look forward to seeing you at one of our future webinars. Have a great day.